With AI and human expertise working hand in hand, we're transforming complexity into clarity and paving the way for better treatments for patients. An immune response happens when different groups of specialized cells coordinate to fight a challenge. And for us, these challenges are diseases where the standard of care isn't enough, and the response is triggered by a novel treatment we are looking into. So to understand this immune response, we need to know which cells are present, in what quantities, and what states they're in. And to gain insight into that, the activity of genes alone or proteins alone aren't enough, you need both. And that's why multimodal single-cell information is so powerful, especially when paired with AI to uncover the hidden features beyond the known markers we are familiar with.
Every cell has a story, and it starts in our New York City lab, where we capture thousands of signals for each cell at once to understand what it is and how it behaves. We barcode and sequence this information, turning its cell's biology into digital code. Then our team in Prague curates and analyzes the data, while our engineers in Israel power the infrastructure that makes this whole pipeline work. By the time it reaches our atlas, each cell has become a digital entity that scientists and AI can learn from. It's a global journey, and it starts with a single cell.
When you look at patient samples at a single-cell resolution, you're not working with averages anymore. Every cell has to be interpreted in its own context. To do this at scale, we've built a pipeline that integrates all the data points for each cell, filters out technical noise, and layers on the biological knowledge. And our machine learning models annotate the cells, and then our immunology experts review and refine these annotations, especially in the edge cases that are harder to define. And it's the combination of data, models, lab validation, and expert insight that ensure that each cell is not only labeled, but is truly understood within the dynamics of the immune system.
To train foundation models that understand immune complexity, you need massive amounts of high-quality data. That's what we've built and continue building at Immunai. Our workflows scale efficiently, reduce variability, and generate reproducible data. Everything flows into Amica, our multi-omic cell atlas, which integrates millions of cells across studies. The shared foundation allows researchers, biologists, and machine learning engineers to work from the same data, uncovering patterns in the immune system that would be impossible to see without this scale. In short, Amica powers the next generation of single-cell AI.
From a single cell to a global data atlas, we're building the most detailed map of the immune system ever created.
With AI and human expertise working hand in hand, we're transforming complexity into clarity and paving the way for better treatments for patients.